Friends, last time we reached at a point where we discussed about the arrival of the eukaryotic cells. This condition came into existence even if there was proper survival and good increase in number of prokaryotic forms. Although the prokaryotic forms were thriving very well, still there was a rise of an arrival of the eukaryotic forms. Hence today we will be discussing about the differences and comparing the two cellular models and discussing as to why and how the eukaryotic models proved to be a better one or not. As of now, first let us discuss in detail the structure of a proper basic prokaryotic cell. These kinds of cells include cells of bacteria, mycoplasm, etc. The principal feature of this kind of model is absence of mainly cell organelles. Now what are cell organelles? All the compartments or parts that are contained inside a cell embedded in a jelly-like substance known as cytoplasm is known as a cell organelle. And prokaryotes are known to have an absence of many cell organelles. Such kind of cells can be found in different kinds of situations. Groups like extremophile bacteria can be found in high saline condition, can be found in hot water springs, in the areas near volcanic eruptions, in forests, in deserts, in colder regions, polar regions, and in basic residential locations as well. One of the problematic areas for this kind of model is error prone mechanism. For example, in case of DNA replication, there is no check over the error in the process of replication. That is, if the DNA copies get error prone, if the base pairs are skipped in reading the DNA and making a copy, then there is no mechanism to recheck it, to correct it, and whatever the pattern the DNA polymerase enzyme detects and contributes in the replication of DNA is followed as a genetic material in the next generation. There is no check or correction measure in such process. Such kind of model, since very basic in structure, does not have many parts, hence requires very less amount of energy for survival. That is why we do not see and witness much of ATP production in case of bacteria and lower forms. One important feature for such kind of cells and few of the prokaryotic form is the presence of an extra chromosomal DNA, which is also known as plasmid. The important characteristic of this plasmid is it is self-replicating in nature. It does not require a specific assembly or a specific armory of enzymes to perform replication. And one of the most important tool in the processes and in the industry of biotechnology, this plasmid DNA has proved to as an important tool in development of vaccines, in development of biopesticides, in development of various chemicals that are useful for human usage, industrial usage, and we can easily develop and we can easily manipulate such kind of DNA material to express proteins and to obtain protein products of our own choice with the help of these plasmid self-replicating DNAs. Now if we compare the model of a basic eukaryotic cell form and cell structure as compared to that of a prokaryotic form, then the first and the foremost difference that we witness and observe is the presence of defined cell boundaries. Cell walls, cell envelopes, cell membranes are specified cell boundaries that define the basic structure of an eukaryotic cell. A eukaryotic cell consists of many subcellular sections, many cell compartments. For example, they have a lot of cellular parts, cellular organelles like mitochondria, nucleus, ribosomes, Golgi apparatus, vacuoles, inclusion bodies, endoplasmic reticulum, etc. Now, such kind of extensive compartmentalization provides one important feature of the eukaryotic cells, and on the basis of which there's a very famous question asked in examinations about the concept of division of labor. Now, what does division of labor means? Labor means amount of work, and division of labor means that in case of eukaryotic cells, different centers and different points are assigned different jobs. For energy production, we have mitochondria. For protein production, we have ribosomes. For calcium storage, we have endoplasmic reticulum. For storage of waste material, liquids, salts, cell sap, we have vacuoles. For packaging and processing of substances, we have Golgi apparatus. Hence, the partition, the parts, and the structure of basic eukaryotic cells proves and justifies the concept of division of labor. Also, a proper eukaryotic cell will have a lot of rectifying mechanisms. Organelles such as Golgi apparatus are known to produce suicidal bags, like suicidal bags known as lysosomes, to destroy and cells that get infected. Eukaryotic cells have different kinds of DNA polymerases, enzymes that are involved in proper copying of the DNA, making copies of the DNA, and if at all there is a skipping of the base pairs, then such process is stopped and restarted. Such kind of cells include modern day human cells, cat cells, paramecium, bird cells, plant cells, etc. Hence, summarizing the differences between prokaryotes and eukaryotes, the first difference is based on design. On one hand, prokaryotes have a simpler design, devoid of many cell parts, and many of them even devoid of cell walls, while on the other hand, the eukaryotic cells are complex. They have different kinds of parts and cell organelles. Each and every cell organelle has a different kind of structure, and there is very extensive and complex sort of compartmentalization, part division, and different parts of the eukaryotic cells. If we consider the work efficiency, if we compare both the models on the basis of the work efficiency, then obviously the prokaryotic cells, since they lack many of the cell parts, so many of the functions are not as properly and as specifically performed by these models. Hence, the number of functions are somewhat counted and limited in case of prokaryotic models. In case of eukaryotic cells, they are multitasking in nature, with each task being taken up by a specific compartment of the cell. They become much more efficient, much more production of substances, energy, much more metabolism of waste product, and hence we witness a multitude of tasks offered by the eukaryotic model as compared to the prokaryotic model. If we compare both the models in terms of their reproductive procedures, then the prokaryotes maximally, it is observed that they reproduce with the help of basic fission or binary fission, but in some bacterial forms, even methodologies like conjugation, transformation, transduction have also been seen, where there is use of plasmodesmata or production of specific kind of particles that get secreted from one strain and communicated to the other strain. While in case of eukaryotic models, two basic sort of cellular processes or divisions are observed to continue with the process of reproduction, that is the mitosis or the equational division, and meiosis or the reductional division. At the end, if we compare the survival instinct and the propagation graph or propagation behavior of prokaryotes and eukaryotes, then prokaryotes, though have a very basic design, lack of lot of parts, still they are omnipresent. They can be found in simple living conditions, still harsh living conditions also, as we already discussed about the extremophile bacteria. While in case of eukaryotic forms, though they are considered to be an advanced form of cellular model, still there is lack of presence of eukaryotic forms in the extreme conditions. Many a times we do not observe the presence of eukaryotic conditions in extreme saline or extreme heat or extreme cold conditions, while this is not the case with the prokaryotic forms. So in a nutshell, if we have to compare and discuss the differences between a prokaryotic model and a eukaryotic model, then we can take up the comparison between a Nokia 3310 and an Android Jelly Bean cell phone. In case of Nokia 3310, we have a keypad and very limited amount of memory and very limited amount of functions, while in case of Android or Jelly Bean, we have a multitude amount of functions, more hardware included, and hence more number of functions included in a Android model. So in this way, at some point prokaryotes are quite successful in certain conditions, while in many conditions eukaryotes are also successful in surviving. Hence in today's time we witness the presence of both the forms, and there is no eradication of prokaryotes from the earth. So friends, here we arrive at the end of our presentation on the topic of fundamental unit of life part 2, and here we witness few questions that students can solve to get more attached to the topic, to get their concepts more clear and to practice a bit. The first question is, why are prokaryotic single cell forms considered immortal? The second is, enlist the major differences in prokaryotic and eukaryotic cells diagrammatically. Question number 3, why can't earliest aggregates like microspheres and conservates be termed as proper cells? Question number 4, what might be the outcome if check mechanisms are absent in higher versions of eukaryotes? And question number 5, if a culture of E. coli, that is Escherichia coli, takes 29 hours to fill half of the culture vessel, then how much time will it take to completely fill the vessel if the doubling rate is 5.4 into 10 into 10 into 10 seconds? Students, kindly solve these questions to get your concepts more correct. In the upcoming part of the series, now we will be picking up individual cell organelles, and we will be discussing the structure, function, and we will sort of justify the functions of different cell organelles in prokaryotes and eukaryotes. Friends, do keep following our channel, subscribe our channel for regular updates of upcoming presentations, and do follow our page on Facebook and Instagram as well. You can also find a page full of biology and other field facts named as BioFact. You can get the link at our FB page. And do keep sending your suggestions and queries and topics on which you would like to see presentations being made. Thank you.